Purine catabolism and gout. Degradation of purine nucleotides. Purines are catabolized to uric acid in human body. An average of 500 to 700 milligram of uric acid is excreted by human beings. Most of it is found in urine. Its normal serum level is 3 to 7 milligram per deciliter. The purines, namely adenine and guanine, is converted into uric acid in human body. The pathway is shown in the slide. The adenosine monophosphate is initially converted into inosine monophosphate with the help of the enzyme adenosine monophosphate deaminase. And the reaction is a deamination reaction. Or the adenosine monophosphate is converted into adenosine with the help of nucleotidase enzyme, where the phosphate group is removed by hydrolysis. And then the adenosine thus formed is again deaminated to inosine, and the reaction is catalyzed by adenosine deaminase. And the inosine monophosphate formed initially can be converted into inosine with the help of the enzyme nucleotidase. And the resultant product, inosine, is converted into hypoxanthine, and the reaction is catalyzed by purine nucleoside phosphorylase. In this reaction, the ribose moiety is removed from the inosine structure as ribose 1-phosphate, and the resultant product is hypoxanthine. The next reaction is catalyzed by xanthine oxidase, where the hypoxanthine is converted into xanthine, and there is the removal of hydrogen peroxide. In the xanthine, there is the incorporation of oxygen and hydrogen at the second and third position of hypoxanthine, and thus it forms xanthine. Similarly, in case of guanosine monophosphate, the guanosine monophosphate is converted into guanosine with the help of the enzyme nucleotidase, where the phosphate group is removed. The next reaction is catalyzed by purine nucleosidase phosphorylase. And here also the ribose moiety is removed as ribose 1-phosphate from guanosine, and thus it forms guanine. And guanine is converted into xanthine, and the enzyme responsible for this conversion is guanase, and the reaction is a typical deamination reaction. Thus, the adenosine monophosphate and the guanosine monophosphate will be channeled to form xanthine, and the xanthine thus formed will be converted into uric acid with the help of the enzyme xanthine oxidase. And the uric acid thus formed will be excreted through urine in normal healthy individuals. The disorders of purine metabolism. Hyperuricemia and gout are the major diseases related to purine metabolism. Hyperuricemia refers to an elevation in the serum uric acid concentration, and this is sometimes associated with increased uric acid excretion also. Now, gout. Gout is a metabolic disease associated with overproduction of uric acid. At the physiological pH, the uric acid is found in a more soluble form as sodium urate. In severe hyperuricemia, crystals of sodium urate get deposited in the soft tissues, particularly in the joints. Such deposits are commonly known as tophi. Usually, this is seen at the foot or in the hands. This causes inflammation in the joints, resulting in a painful gouty arthritis. Sodium urate or uric acid may also precipitate in kidneys and ureters, thus that results in renal damage and stone formation. The picture below shows the gouty arthritis, tophi, accumulation of crystals of sodium urate at the joints. The X-ray image below shows a normal foot and deposition of sodium urate crystals at the toe. Gout is a chronic disorder. It is characterized by excess of uric acid in blood, hyperuricemia, and deposition of sodium monourate in alveolar and non-alveolar structures, producing the so-called tophi. There are two types of gout. First one, primary gout. Second one, secondary gout. First one, primary gout. Here the hyperuricemia is not due to the increased destruction of nucleic acid. The essential abnormality is increased formation of uric acid from simple carbon and nitrogen compounds without intermediary incorporation into nucleic acids. And the primary gout can be primary metabolic gout or primary renal gout. In case of metabolic gout, it is due to inherited metabolic defect in purine metabolism, leading to excessive rate of conversion of glycine, the amino acid, to uric acid. In fact, the glycine is one of the major amino acid that is used in the synthesis of the purines. Second one, primary renal gout. It is due to the failure in the uric acid excretion through the renal system. Secondary gout. The secondary gout can be secondary metabolic gout or secondary renal gout. In case of secondary metabolic gout, the disease is due to secondary increase in purine catabolism in conditions like leukemia or prolonged fasting and polycythemia. In case of secondary renal gout, it is due to defective glomerular filtration of urate due to generalized renal failure. In case of von Gerke's disease, the glycogen storage disease, there is a deficiency of glucose 6-phosphatase, and this leads to elevated rate of pentose formation in hexose monophosphate shunt. This acts as a good substrate for PRPP synthetase or phosphoribosyl pyrophosphate synthetase and enhances the synthesis of purine followed by their catabolism to uric acid. Increased lactic acid competes with uric acid excretion, resulting to retention of uric acid, thus causing hyperuricemia. Treatment policies in gout. The first and foremost policy in treatment gout is restriction of alcohol and reduced dietary intake of purine containing food, especially red meat or organ meat. Second one, increase renal excretion of urate by uricosuric drugs, which decrease the reabsorption of uric acid from kidney tubules, example probenecid. Third one, reduce urate production by allopurinol, an analog of hyperxanthine. Allopurinol is the competitive inhibitor of xanthine oxidase, thereby decreasing the formation of uric acid. Fourth one, colchicine, an anti-inflammatory agent, is used to arrest the arthritis in gout. Fifth, use of PEG uricase or polyethylene glycol urase combination and conversion of uric acid to allantoin, which is a more water soluble and easily excreted form, is being tried as a therapeutic measure to reduce body uric acid pool. The diagram below shows the impact of allopurinol, which blocks the formation of uric acid from the purine. The next shows the uricase. It breaks down the once formed uric acid. The probenecid, this increases the excretion of uric acid in urine. Thus, it minimizes the deposition of crystals. Non-steroid anti-inflammatory drugs, colchicine and corticosteroids. This can reduce inflammation when gouty arthritis is present. Thank you.